Not saying okay. I am not saying okay. I'm saying, I'm not sure what I'm saying, but let's get going. Very excited about biotechnology, DNA biotechnology. All right. So let's first look at, well, I think no, we didn't. Okay. So genetic engineering. This is a very, very important part of genetics, area of research. Field. This is a field of silence. It's not. It's a field of science that deals with manipulating the genome. So manipulating the DNA, that's what genetic engineering does. So when we talk about recombination, recombinant DNA is the major focus of this genetic engineering. So this is where DNA from two different sources is joined into one molecule. So in this case, you have a bacterial chromosome and then you have a plasmid that's going to be joined together. It's going to be recombined. All right. And let's see. I don't know if this is, no. All right. So, so it's going to be, so if we look at this process here, we have this bacterium, we have the bacterial chromosome, and then the plasmid. We have the recombinant DNA. You can see we isolate the plasmid DNA and the DNA containing the gene of interest. And the gene is inserted into the plasmid. So we take out a piece of this, and or you can see right here, I'm not sure why that's empty, but so we take out this little piece here and we insert it into the plasmid, and then the plasmid is put back into the bacterial cell, and the cells are cloned that have this gene of interest in it, and it identifies some sort of desired clone. So you can see the gene for pest resistance is inserted into plants. The genes used to alter bacteria for cleaning up toxic waste. So bacteria that would be good at cleaning up that particular toxic waste. The basic research on proteins, human growth hormone that treats people that have stunted growth. We can do genetic recombination with that. Proteins that dissolve blood clots in heart attack therapy. I mean, there is just unbelievable, and there's so much that we wouldn't even know. I mean, when you eat strawberries that are really, really sweet and large, that's not how strawberries naturally are. And so there's been genetic modification going on there. All right, so cutting the DNA sources using a restriction enzyme. So there's an enzyme that we use that restricts and cuts. So you can see this is the restriction site. And this is when we're inserting a single gene into the bacteria. And so it's going to, the restriction enzymes cut DNA at a specific site, and that site is referred to as the restriction site. And then you have your genes of interest, and they could be antibiotic resistant genes, excuse me, are cut into what's called resistant fragments. So we use these restriction enzymes. Then when the DNA is cut, we use these sticky ends, or these single-stranded DNAs. Using these restriction enzymes, they create these stick ends onto the DNA molecule. So when the DNA is cut, those sticky ends or single-stranded DNA are created. So the same restriction enzyme has to be used on both the plasmid and the DNA donor source. And the sticky ends of the plasmid and the genes of interest are going to match, and then they can be joined together. So they have to be the same. So you can see the DNA fragment produced by the same restriction enzyme. So we have this sticky end, and you can see this fragment right here, and then you can see how the G comes in and fits here, and then the G from here. So you can see, and then here the G from this one, and then here's this sequence right here, and then with the G ending up there, and then we go into that sequence there. All right, then of course you have DNA ligase that seals these strands, seals them together, and you end up with this recombinant DNA molecule in the end. All right, so when we're combining DNA from both sources, those sticky ends have to match. So you can see if we put those together, they would match. They'd also match the new piece that we're adding into it. Those have to be in the same shape. So insert the human DNA into the plasmids, and then you're going to mix them together. All right. So, and then we reintroduce the recombined plasmid. Like it said, this is just a blown up picture of what we saw. We reintroduce it back in here, and then you let the bacteria grow and reproduce. It's going to make lots more copies, but now it's going to make copies with that new material that's added in. This is how my neon fish, that's how they were glowing, is because they took the glowing material from jellyfish and they inserted it into the DNA of the fish, and they were able to insert the glowing portion into the fish. All right, so here is the whole process of cloning. Again, step 1, the restriction enzymes. Step 2, create the condition for the bacteria to take up the recombined plasmids. Now the bacteria are now transformed. So now we have this transformed bacteria. And then we incubate them. We put the plasmids in, we incubate them, and we grow them up. And then you end up with the transformed bacteria. And they reproduce through this process of binary fission. So it's asexual reproduction. You're going to get an exact copy. You're not mixing any genetic material here. And the outcome is we end up with transformed cells that have antibiotic resistance. So if we're trying to make antibiotic resistant, so here you have ampicillin resistant. Again, ampicillin, antibiotic. So this is, and this is how ultimately we end up with antibiotic resistant genes. Whoops. We already looked at using this. I'm not sure. Oh, Carrie Mullis. So when we talk about this process, this polymerase chain reaction, this is a process that requires no organism in the production of new DNA molecules. So Carrie Mullis won, this gentleman here, won the Nobel Prize in 1993 for the development of this process. And this process is used to turn a single molecule of DNA into a large workable sample of 100% identical DNA. So this DNA is exactly 100%. Well, here it's 100% identical to that original one. Now this one is widely used in criminal forensics cases. This is an unbelievable process now in genetics research, because they can make you can make so many copies of the DNA and do so much research so very, very quickly. So what happens in this process of we call it PCR. So because you're not going to keep saying polymerase chain reaction, we just call it PCR. So the DNA sample is placed in a PCR thermal cycler. Thermal means heat, and so you can see here heat. We're going to heat this DNA up briefly to separate these DNA strands. So you heat them up and it's going to separate those strands. The machine uses heat. It's going to use DNA primers. It's going to use these primers. It's going to use enzymes, polymerase. It's going to use enzymes and a constant supply of nucleosides. So it's going to have lots and lots of them in order to build the new DNA that are going to be identical. All right, so we heat them up so that heat up the DNA so replication can occur, and then we're going to attach the primers. All right, so the attachment of the DNA primer to the DNA template strand is going to start the replication. Okay, so the DNA polymerase adds nucleotides to the 3' end of each of the primers. All right, so the 3' end there. The DNA polymerase enzyme works, again, 5' to 3', attaching the nucleosides to the growing new side of the DNA, of the replicated DNA. Now, so We've split it up, we've heated it, split it up, added primers, added the nucleotides to it, and now we're going to cool it off and we're going to glue them back together. Okay, so we cool it off to recombine and stabilize the DNA back into the double strands, and we're going to repeat that cycle over and over and over. And then we perform a gel electrophoresis test to separate for a comparison of the nucleotide sequences between organisms. And so that's PCR. So we start off with one piece of DNA, and we have to make lots and lots of copies because we have to run a lot of tests. Think about it. In a police case, you're going to want a lot of evidence, and sometimes there's not very much. And so you've got to be able to make copies of that DNA in order to make that happen. That's all, folks. Yeah.